Hello, I'd like to recap the last couple of weeks that you have been in biology because a lot has gone on. My hope is that I'm going to be able to connect the two chapters that you have been responsible for going over, taking notes on, and studying. That would be chapters 1 and chapter 6. Chapter 1 is going to introduce you to the scientific method. And in the scientific method, you learned about experimental variables, controlled variables, how to make a data table, etc. in class. Also, it introduced you to the whole classification and idea of classification of living things. We have not yet gotten to that part yet, because what I did is I jumped over to chapter 6, and I jumped over to chapter 6 because it introduced you to chemistry. And in the introduction to chemistry, hopefully you're able to get an idea of what chemical bonds are, from ionic bond to a covalent bond to even a hydrogen bond. But more importantly, how elements, in particular those CHNOPs, make up most of the living things that we are going to be studying. The other thing that we have to realize is that not just these elements right in here, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, but magnesium is important, sodium is important, chlorine is important, fluorine. The different salts and the different metals are also going to be important to living organisms, if not on a trace element level, at least involved with respect to some of these concepts that we're going to go over, such as osmosis and osmoregulation. In the process of science, you've done some controlled experiments. Let's say, for example, that of the murder mystery of Chester. You always were able to design things very similar. So in other words, when you tested the powders and the pH, you did a controlled experiment. How you did that controlled experiment is that you put each of those powders into a solution of water, approximately 10 mils of it, and then you tested it with the same pH paper. So because you were doing the same things for these different powders, you were controlling the experiment. You did not know the outcome or the result of the pH, because the pH was going to be dependent upon which powder was being tested. But the idea is that you were indeed doing controlled experiments while you were trying to solve Chester's murder mystery. You were also doing controlled experiment during the unknown. And during the unknown organic molecule, which varied from something as simple as some glucose that I mixed up with water to as complicated as a sardine mixed along with some starch. I'm sorry for those of you who got it. It was pretty gross. But the idea is that you were also doing a controlled experiment. You had 2 mils of the solution. You were using the known reagents and how they react, how Benedict's reacted, how biurettes and how iodine reacted with their specific compound. So by doing all of those types of controlled experiments, you're able to eventually get to the unknown and identify what type of molecules were in that unknown substance that was in the beaker. So you have been doing this on the side. So, and the other thing is, in theory, we could look at these different types of controlled groups. We always have the control groups and the experimental group. Any sort of design of an experiment has to have 2 groups. Just like the multiple choice in that first quiz that I gave you, it made no sense to have a scientific experiment with only one group. One group always has to have that experimental variable. So you'll notice there's a difference between a group and a variable. These are groups or subjects in numerical times. For example, the control group of the mice with a sweetener. You could have a whole bunch of numbers of mice that are not receiving the actual sweetener, and then you could have the experimental group, or the number of individual mice which are receiving the sweetener. So there's a difference between the group and the variable, but it gets very confusing because there are often you might read that they might be subtly interchanged. But think about the group as a number of individuals or number of organisms that is being tested. So in these 2 labs here, I love them because I thought they were probably a really good introduction to the scientific method, and they're called the inquiry-based lab. You weren't quite sure what was going to happen. They were somewhat open-ended, but yet they're also guided in terms of what the results would be. So, for example, with Chester's murder mystery, you were given the alibis. Ultimately, you were doing the test, and then you were going to do the collection of data. You also did the collection of data in the organic molecules. And like any good science, the data will help you lead to the conclusion. But your conclusion is irrelevant in science unless it is backed up by the data. So we did want to spend time on how to collect data, what types of data there were going to be available. So in any case, the data, remember, will always support any type of conclusion. And that's one of the things I looked for in reading the conclusion of Chester's murder mystery. And we have also gone over the fact that there are two main types of data. The quantitative, think about that which is numerical. Whether it would be the BMI of the mice, whether it would be, for example, the time or the temperature of the day according to time, so that the data that is quantifiable is numerical. We've also gone over the fact that in the types of data, we now get something, the dependent variable versus the independent variable. By now, you should have it pretty much solidified that on the y-axis, you are going to have the dependent variable, that which is going to respond to however the design of the experiment is set up. The independent variable is going to be that on the x-axis. For those of you who did your graph wrong, it should have looked kind of strange when you plotted the data and you got the points going like that, rather than as more like expected, up like that according to the bubble. So anytime you see a weird looking graph, that's kind of an indication that you are mixing up your variables. So remember, the x-axis is going to be that which you as a scientist can set up. You can set up, for example, how often you want to collect data. Is it once a year, once an hour, once a minute? Or do you want to collect data at different pHs? Or do you want to collect data at different temperatures? You are controlling that group of data that you are now going to consider to be the independent, because it will not respond to the dependent. It's the reverse. The dependent responds to how you set up the data. The other type of data that we looked at is qualitative. Much of Chester's murder mystery was about this. We were looking at, for example, the color of the reagent, the smell of the burning fabric, the idea of the chromatography and how the pen gave that different color. Again, we're back to color. And even the reagents of the unknown, we're looking at the color change. So much of what we did the first couple of weeks was that of qualitative data. A little quantitative data is something that we're going to look at for other experiments, but hopefully you have a really good hand of what qualitative data is. So in this hierarchy of biology, elements are significant. We need to know that of hydrogen, oxygen, how those are made up, and more importantly, it's these electrons that are going to be involved anytime a molecule is going to be formed. We're also going to see something that in biology is often called an emergent property. The emergent property is, in general, think about how chlorine in and of itself is a gas and highly dangerous, how sodium in and of by itself is a soft metal and highly reactive, but when these two combine, we get something that's very flavorful called table salt. So essentially, chlorine and sodium have emerged by joining together. They now have a brand new property that we can now consider table salt or sodium chloride. So we're going to see how these molecules then bring on different characteristics. And ultimately, the molecules, we're going to see how the molecules form cells from the outer membrane, which is made up of phospholipids and proteins, and ultimately how these cells make up the organism. So the reason why we have spent a lot of time on chemistry is to get a really good basis of how these elements respond, because we're going to see them over and over again as we study living organisms. And by now you should have a really good idea of the types of bonding, the difference between an ionic bond in which the electrons are transferred, or a covalent bond in which the electrons are shared. In either case, whether the electrons are transferred from the outer orbital or whether the electrons are shared, both of these cases are in order to make a more stable element. Okay, sorry about that. The last thing not last thing, but the next little part of chapter 6 is that of water. The chemistry of water. Water is going to be probably one of the most important molecules in our particular bodies. 70%, 80% of our cells are made up of water. And the reason is that water is really unique in terms of its ability to form these hydrogen bonds. And these hydrogen bonds are going to be able to give water its main properties. The properties of water that we're going to review: adhesion and cohesion. That is going to be significant for those who know about plants and how plants are able to get water from its roots all the way up through the leaves through a special type of tissue called xylem. Adhesion and cohesion is also significant in terms of our arteries, veins, and capillaries and keeping that blood flowing in the plasma. Then we're going to get surface tension. Surface tension is going to allow for some things to float on water. It's not that important, except, of course, if you're a water strider and don't want to cross it. But surface tension is something that all of us has experienced. Surface tension, if you've ever done a belly flop in the swimming pool, you have experienced the surface tension. Literally, it forms a film. The water molecules form a film that is as strong as almost concrete, depending on how high you jump. The other thing we're going to see is that, this is pretty cool, solid is less dense. I always like to think of it as ice floats. You think if you were an organism in a pond, you were very grateful that the ice is floating rather than sinking down. It is unique in terms of all the molecules. Most molecules in their solid form are denser than in their liquid form. Ice is different. Ice is less dense in its liquid form. The other thing that we're going to see is that the near universal solvent. For all those things that I mixed up in the beaker, I used water. And you notice, if you were some of those lucky souls that may have gotten the Twinkie or the potato chips or the bacon, you saw that there is often a layer of this lipid that did not commingle with the water. But other than that, from the donuts to the glucose to the baking soda to the salt, I was able to use water to dissolve all these different types of chemicals. Oops. Pause. Okay, a little technical difficulty. I must have pressed the wrong button. Okay, so in that part here, the universal solvent, and then also water is going to moderate the temperature, not only of our body, but of the earth itself. So from a living organism level, for example, when you're exercising and sweating, these hydrogen bonds are broken. By being broken, they're going to carry off a lot of heat energy, and that is what will allow you to cool down. Same thing is, for example, those of us living along the California coast or the East Coast or any area that's surrounded by large bodies of water, that heat is given off slowly and absorbed slowly, thus keeping temperatures of the air relatively mild in comparison to land that is surrounded by even more land. So here's a pretty good depiction of how water is going to be a solvent. It's because of this polar ability of water to have these two charges. For example, here, sodium is a plus charge, and the water oxygen, if you remember, the oxygen in the water is a negative charge. So that way there, notice how the water oxygen molecules orient themselves around the sodium. Conversely, when you look at the other picture of the chlorine, chlorine is negatively charged, and you're going to notice is that the hydrogen atoms are going to be surrounding that of the chlorine ion. So by being polar, this water has the ability to dissolve many different substances, such as sodium chloride. It can also dissolve other larger molecules, for example, in terms of a protein. So you could have smaller protein atoms or molecules surrounded by water in a different solution. But because we're looking at something very different, the protein is being dissolved, not digested. If it were being digested, that would be very similar to a hydrolysis reaction. So there's a difference between, for example, the protein that was in the sardine or the protein that was in the bacon being surrounded by water, thus being able to be tested by biurettes, compared to being chemically digested by an enzyme. And hopefully you by now have this down pat in terms of polymers to monomers. One of the things we did with the Nearpod reaction is we saw that, okay, Nearpod activity I should say, that if we can represent all the monomers as single units, if, for example, these single units were to be put together by this process called dehydration synthesis, in which water is removed, we can ultimately form a polymer. Subsequently, these polymers can be broken down by another process called hydrolysis, in which H2O is added to a chemical reaction. Regardless of whether it's going to be hydrolysis or dehydration synthesis, neither could occur unless there are enzymes that were going to either put the molecules together or break the molecules apart. So by now you should have it pretty solid what a monomer is, and you should have it pretty solid what a polymer is. And so lastly, what we're looking at, we have spent a lot of time down here at the molecular level. And hopefully by understanding how chemical bonds work, how chemical elements work and react in order to form different bonds, whether it's an ionic bond or covalent bond, we are now going to take the next step and start learning about cells and how cells are going to keep a homeostatic environment because they are able to have a plasma membrane made up of phospholipids and proteins, which are on the molecular level, and how these cells can indeed be unique in terms of whether they are unicellular organisms such as an amoeba or whether they are a group of cells joining to form a tissue. So hopefully by getting a better understanding on the molecular level, as we look into cells, we're also going to look at how these molecules evolved into the cellular level. Okay, and that's going to be what our next little chunk of information is going to be. And on the cellular level, we're looking at Chapter 7. I'll give you more information on that on our agenda later on.